In the stillness of night, as your breathing slows and your body begins to relax, let your mind float gently into the world of life science, where quiet strands of molecules carry messages written millions of years ago, silently shaping who we are. Genetics is, in essence, the story of life, written in code, a language not spoken aloud but inscribed within every cell of every living thing, from the tiniest microbe in a drop of pond water to the tallest tree stretching skyward in a silent forest. It is a language without sound, without punctuation, a language carried in sequences, in structures, in the careful pairing of molecular letters. This part of our journey begins not with a loud discovery, but with something far softer, the idea that within you, right now, lies a complete set of instructions, instructions for building you, instructions for sustaining you, and instructions, too, for passing pieces of yourself onward to future generations, as life always has and always will. These instructions are housed in an extraordinary molecule called DNA deoxyribonucleic acid, though names hardly matter when the story is this gentle. DNA is not sharp or harsh. It does not shout. Instead, it flows in long, spiraling strands, like the soft curls of a ribbon on a breeze. Its shape, the famous double helix, is elegant and quiet, two delicate threads winding around each other in a graceful dance of balance and symmetry. This DNA resides inside the cells of your body, not just some of them, nearly every single one, whether it's a muscle cell in your leg or a nerve cell in your brain. Each carries the complete instruction manual for your entire being. And yet, remarkably, most of those instructions are folded away, resting. Only the portions needed for each specific task are gently read and followed. Imagine, if you will, a vast, silent library, one tucked inside every microscopic cell. Its shelves are filled with volumes written in a script only the cell can interpret. Some books are opened and read regularly, guiding your cells to make proteins and carry out daily work. Others remain closed for years, perhaps a lifetime. Some may never be read at all. Yet there they rest, carefully preserved, in case the need ever arises. When we speak of heredity, the passing of traits from parent to child, we are speaking of this very library being duplicated and delivered, one generation after another. Each of us receives a copy of these instructions from both of our parents. Half of our genetic material comes from one side, and half from the other, carefully mingled together in a unique pattern that will never repeat exactly again. This pattern is what makes you you. It determines the shape of your hands, the pitch of your voice, the texture of your hair. It even gently influences how your body responds to the world around you, how quickly your muscles recover, how your immune system reacts, how your eyes perceive the soft colors of dawn. All of this, and so much more, written in molecular letters, too small to see, yet vast in their meaning. And it's not just humans. The same quiet code shapes the hummingbird, the sunflower, the coral reef, the elephant. Every living creature carries its own version of the script, adapted for its needs, yet following the same basic rules, rules that have guided life on Earth for billions of years. When you lie down to rest, your cells do not sleep. Within you, gentle processes continue. Some genes quietly activate, producing proteins needed for repair and maintenance. Others turn off for the night. Hormones shift, tissues rebuild. Even in sleep, the language of life is being read, interpreted, and carried out with perfect calm. The beauty of this process is not just in its detail, but in its harmony. Nothing in the cell works alone. Just as a forest thrives not because of a single tree, but because of the ecosystem as a whole, so too does each cell thrive through cooperation. Proteins carry messages, enzymes perform tasks, molecules drift through the cytoplasm like boats on a slow-moving river. Everything has its place. Everything has its rhythm. And so, when we begin our study of genetics and heredity, We are not diving into something loud or complex or intimidating. We are stepping into a world that already knows how to be quiet, a world that has existed in every organism since the beginning of life, a world of patterns, repetition, and gentle variation. Over the course of this lecture, we will walk softly through that world. We'll explore how genes work, how traits are inherited, and how cells pass their knowledge forward. We'll visit Mendel's peaceful gardens, where pea plants first whispered their secrets. We'll watch as chromosomes quietly align and separate in perfect formation. And we'll look closely at the way life changes, not in leaps and shouts, but in slow, spiraling motions. But for now, as we rest in this first chapter, let yourself simply reflect on the idea that you are made of stories. Stories written in code. Stories older than your name, older than your family, older even than your species. These stories are not loud. They are not boastful. They are quiet, careful, and kind. And like the turning of the earth or the rhythm of a heartbeat, they continue silently, gently, even as you sleep. To what is genetics? A quiet definition. As your breath continues to slow and the weight of the day begins to lift, we ease a little deeper into the gentle world of life science, where knowledge is not rushed and every idea unfolds like the soft petals of a blooming flower. Genetics, at its core, is the study of heredity, but more softly, it is the science of connection, of how traits, tendencies, and possibilities are carried from one generation to the next. It is a study not of noise or spectacle, but of the subtle, unbroken threads that link parents to children and organisms to their ancestors, stretching far back through time. To understand genetics is to understand how life remembers. Every living organism, from a moss-covered stone in a quiet forest to the most ancient of whales, gliding through the sea, carries with it a complete record of its biological inheritance. That record is not stored in books or photographs or maps, but in molecules. Specifically, in DNA, the silent spiral of nucleotides we met earlier, a molecule that preserves information with breathtaking precision. The word genetics comes from the same root as generation and genesis, words that suggest birth, origin, and continuity. It refers to the patterns by which traits emerge, develop, and carry on. A child with blue eyes, for instance, does not acquire them by chance. Those eyes are the expression of genetic instructions passed down from ancestors, perhaps a mother, a grandfather, or someone farther back still. And yet, genetics is not just about visible traits. It also quietly shapes how the body functions. It influences how cells grow and divide, how enzymes operate, and how the immune system responds to the world. From the curling of fingers to the digestion of food, the work of genes unfolds silently in the background, an unspoken symphony conducted by tiny molecules within the cell. In classical genetics, the kind first explored by Gregor Mendel in the 1800s, traits are inherited according to predictable patterns. Mendel, a monk with a quiet mind and a careful hand, tended to pea plants in a serene garden. He noticed how certain traits like flower color or seed shape passed from one generation to the next in mathematical proportions. Through many seasons and patient observations, Mendel uncovered some of the fundamental rules of inheritance. He discovered that traits are controlled by units we now call genes, and that these genes come in pairs, one from each parent. Some versions of a gene, known as alleles, are dominant, meaning they tend to be expressed more visibly. Others are recessive, quietly waiting in the background, often only showing themselves when both copies are the same. This basic framework of inheritance, dominant and recessive alleles, passed down in predictable ratios, is now known as Mendelian genetics. And while not all traits follow these simple patterns, Mendel's work laid the soft foundation for everything that came after. Today, the study of genetics extends far beyond pea plants. It includes the exploration of the entire genome, all of an organism's DNA. For humans, this means over 20 genes quietly arranged across 23 pairs of chromosomes. Within this vast library of instructions, some genes are well understood, while others remain softly mysterious, their functions not yet fully known. Modern genetics also explores the structure of DNA, the mechanisms by which it is copied and repaired, and the ways in which genes are turned on or off, a process known as gene regulation. These discoveries help explain why, even with the same set of genes, different cells in your body behave so differently. A muscle cell and a nerve cell both carry your full genome, yet each follows a different script, producing only the proteins it needs. The field of genetics has also given rise to new branches like molecular genetics, which studies the chemical structure of DNA and how it replicates, or population genetics, which looks at how genes spread through groups of organisms over time. There is behavioral genetics, exploring how inherited tendencies may influence how animals act, and epigenetics, which we'll visit later, a field examining how experiences and environment can affect gene expression without altering the DNA sequence itself. In essence, genetics is not just about what is passed down, but also about how it is passed, why, and in what combinations. It is the quiet study of biological storytelling of how each generation receives, rewrites, and relays the tale of life. It is important to remember that genes do not dictate destiny. They offer possibilities. A child may inherit a genetic tendency toward musical skill, for example, but without the opportunity to practice or hear music, that potential may remain unrealized. In the same way, a genetic vulnerability to illness may never unfold if balanced by healthy habits and environmental support. The story of life is shaped not only by the text, but also by the setting in which it is read. As we gently explore further into this world, we will encounter terms like genotype, the internal, genetic makeup of an organism, and phenotype, the outward expression of that makeup, shaped by both genes and environment. We will see how tiny changes in DNA called mutations can sometimes lead to new traits or conditions. We will explore how cells use RNA to read DNA's instructions, and how proteins quietly carry out the body's needs. But for now, it is enough simply to know that every living organism is shaped by inheritance, by a silent transmission of information stretching back through uncounted generations. Whether it is the shape of a leaf or the curl of an ear, a sleeping cell or a waking thought, life unfolds according to instructions that were given long before. And in this way, genetics is not just a science. It is a form of memory, a gentle remembering of where we came from, of what we are made of, and of what we might yet become. As your eyes grow heavy and your mind drifts further into rest, let yourself settle into the comfort of continuity, the quiet knowledge that within you lies a story, carefully written, endlessly preserved, and passed forward, even as you sleep. 3. The history of heredity. From gardens to jennies, let us now drift further back in time, not to the beginning of life itself, but to the beginning of our understanding of how life remembers, a time before microscopes or molecular biology, a time when the deepest secrets of heredity lay hidden in the petals of flowers and the shape of seeds. A time when one man, in quiet solitude, began to notice something extraordinary. Our journey begins in the mid-1800s, in a monastery garden, nestled in what is now the Czech Republic. The man tending to the plants was not a famous scientist, nor did he set out to change the course of biology. His name was Gregor Mendel, and he was an Augustinian monk. His days were filled with prayer, study, and the careful cultivation of pea plants. A simple life, lived slowly and with purpose. What Mendel noticed, over the course of many seasons, was that traits in his garden did not appear randomly. Certain features like the color of the pea flowers, or the texture of their pods followed a pattern when passed from parent plants to their offspring. These patterns were subtle at first, almost invisible, but through patient record-keeping and methodical breeding, Mendel uncovered what would become the foundational principles of modern genetics. He worked with Pisum sativum, the common garden pea. This unassuming plant offered clear, distinct traits, flowers that were either purple or white, seeds that were round or wrinkled, and perhaps more importantly, pea plants could be easily cross-pollinated by hand. Mendel would remove the pollen-producing parts from one plant, then brush pollen from another onto it, carefully recording the results across many generations. Through this process, he discovered that some traits would disappear in one generation, only to reappear in the next. A white flower might vanish when two plants were crossed, but then show up again in their grandchildren. Mendel realized that traits were not blended or diluted, as was once believed. Instead, they were carried like small, indivisible units, now called genes. He proposed that each trait was controlled by two factors, one from each parent, what we now call alleles. If both factors were the same, the trait would be pure. If they differed, one might dominate the other. This gave rise to his now famous principles. The law of segregation, which describes how genes separate during reproduction, and the law of independent assortment, which explains how different traits are inherited independently of one another. Mendel didn't use the word gene. That concept would come decades later, but the essence of his insight was remarkably clear. He had discovered that traits are inherited according to predictable rules that could be described mathematically and tested through careful observation. This was the birth of classical genetics. At the time, however, Mendel's work went largely unnoticed. It was published quietly in 1866 in a little red journal and was not recognized for its significance until long after his death. The scientific world, focused on visible anatomy and grand theories of evolution, simply wasn't ready to hear what a monk and his peas had to say. It wasn't until the early 1900s that three independent researchers working in different countries stumbled upon Mendel's findings and realized their value. His work was finally brought to light, and the simple monastery garden became the symbolic birthplace of modern genetics. From there, the story continued to unfold. As technology advanced, so did our tools for studying life. In the 20th century, scientists discovered that genes are made of DNA, and that this DNA is housed within the chromosomes inside the cell's nucleus. They learned how DNA is replicated, how it directs the creation of proteins, and how it can be mutated, leading to variation and sometimes illness. By 1953, two researchers, James Watson and Francis Crick, proposed the now famous double helix structure of DNA. With its ladder-like shape spiraling gently through space, it became an icon of life science. Their discovery, informed by the careful imaging work of Rosalind Franklin, marked a turning point transforming genetics from a garden observation into a molecular science. From the monastery garden to the molecular lab, the story of heredity had become clearer. In recent decades, the pace of discovery has only increased. The Human Genome Project, completed in the early 2000s, mapped all the genes in the human DNA sequence, over 3 billion letters long. Genetic technologies now allow us to understand inherited conditions, trace ancestry, and even edit genes with tools like CRISPR, a technique inspired by bacterial defense systems, as quiet in its origins as Mendel's peas. And yet, even as the science becomes more complex, its foundation remains the same: the passing of information from one generation to the next, the gentle transmission of traits, the unfolding of life according to ancient instructions, handed down without fanfare. The beauty of Mendel's work, and of genetics itself, lies in its clarity. that from something as simple as a seed, we can learn about the deepest workings of life, that through patterns and probabilities, we can glimpse the quiet logic beneath the surface, that within every living thing, from garden plants to human beings, lies a coded history written not in words, but in base pairs and sequences, unchanged by time and untouched by noise. Tonight, as you rest, imagine that garden, the soft hum of insects, the rustle of leaves in the afternoon breeze, and a quiet figure moving among the rows, gently collecting pods, counting seeds, and writing down observations. There are no machines, no microscopes, only patience, curiosity, and a mind open to the rhythms of nature. From such stillness, the science of heredity was born, for the cell, home of the genetic blueprint, beneath the surface of your skin, within every tissue and organ, lies a world so small it escapes the eye, yet it is within this world that all of life's quiet planning unfolds. This world is made of cells, the fundamental units of life, and within each cell, in a place of remarkable stillness and structure, resides the genetic blueprint that guides your body's every moment. The story of heredity cannot be told without first understanding the home in which it lives. The cell is more than just a container, it is a carefully balanced ecosystem, a chamber of molecular activity, and a sanctuary for DNA. There are many kinds of cells. Some are single, free-living organisms, like the tiny bacteria that drift through water or dwell in the soil. Others, like the cells in your body, are part of a larger whole, millions upon millions of them, working together in silence. Whether alone or among many, all living cells share some basic features. Each is wrapped in a delicate membrane. Each carries out essential processes, and nearly all of them, especially in complex life, carry the same precious cargo, a full set of genetic instructions. Let us gently step inside a typical eukaryotic cell, the kind found in humans, animals, plants, and fungi. Picture a softly rounded structure, perhaps shaped like a tiny droplet of water. Suspended within it is a clear jelly-like substance called cytoplasm, a medium that cushions and surrounds the internal components. Floating in this calm interior are structures called organelles, each performing a specialized role. But at the center of it all, often in a slightly denser region, we find the nucleus, the cell's central archive, its control room. The nucleus is enclosed in its own membrane, gently separating it from the rest of the cell. It is here, in this quiet core, that your DNA resides coiled, compacted, and protected. Inside the nucleus, DNA is organized into chromosomes, thread-like structures made of both genetic material and proteins. In humans, there are 23 pairs of chromosomes, for a total of 46. One chromosome of each pair comes from your mother, and the other from your father. Together, they carry the blueprint for your entire body. But DNA is not a rigid script. It is dynamic, unwinding, and rewinding as needed. When the cell is at rest, DNA stretches out in long, loose strands called chromatin, making it easier to access and read. When the cell prepares to divide, these strands condense into the distinct chromosomes familiar from textbook images, the graceful X-shaped forms that appear in diagrams and under powerful microscopes. The instructions encoded in DNA are not read all at once. Instead, the cell selects which genes to use based on need and environment. This reading process begins with a gentle copying of the genetic message into another molecule, RNA. This temporary messenger carries the instructions out of the nucleus and into the surrounding cytoplasm, where another organelle awaits, the ribosome. Ribosomes are the cell's quiet builders. Using the RNA as a guide, they assemble proteins, the true laborers of life. These proteins might become enzymes, helping to speed up chemical reactions, or structural proteins, forming the fibers in skin and bone, or regulatory molecules, orchestrating growth, repair, and communication. And all of this, every strand of DNA, every copied message, every protein produced, occurs inside a space so small that millions of cells could rest on the tip of your finger. Within the cell, balance is everything. Nutrients are taken in, waste is removed, and energy is carefully distributed. The mitochondria, sometimes called the powerhouses of the cell, convert fuel into usable energy, not with noise or combustion, but through slow, precise chemical reactions. The endoplasmic reticulum and Golgi apparatus fold, process, and transport proteins. The lysosomes quietly dismantle and recycle unneeded materials. Each organelle plays its role without rush or competition, moving in harmony, bound by the logic of life and the guidance of genes. Even the cell membrane, the outer boundary, is more than a wall. It is a communicator, sensing the world outside and allowing selected molecules to drift in and out like visitors to a sacred space. And as the cell grows and eventually divides, it must copy its DNA gently, accurately, completely. This process, called DNA replication, ensures that each new cell receives the full set of instructions. It is not fast or chaotic. It unfolds in a calm, methodical sequence, with proteins unwinding the double helix, reading the strands, and forming two identical copies, ready to be passed on. In this way, the cell is not just the home of the genetic blueprint, it is also the reader, the interpreter, the caretaker, and the messenger. It protects the instructions it carries, it follows them, and it passes them onward in a cycle as old as life itself. It is humbling to think that your entire body, from the tips of your eyelashes to the arches of your feet, is made of cells, that every thought, movement, and sensation begins as a microscopic collaboration within these silent rooms, and that inside nearly every one, your DNA waits quietly, ready to guide, to build, and to remember. As you relax deeper into sleep, imagine drifting down to that cellular level. Picture yourself floating gently in the cytoplasm, surrounded by the hum of organelles, the quiet flicker of energy conversion, the slow winding of DNA. There are no sharp edges here, no loud signals, only the soft rhythm of life carrying out its timeless instructions. Here, in the heart of the cell, the story of heredity continues, with no need for words, with no need for sound, just a sequence of letters, folded in silence, waiting patiently always to be read. 5. DNA, the whispering helix. As the night stretches onward and your breath falls into its natural rhythm, let us turn now to the quietest of blueprints, the molecule at the heart of life's continuity, a molecule so small, so delicate in form, and yet so complete in its capacity to hold the secrets of existence. This is DNA, or deoxyribonucleic acid. Though the name may sound complex, the reality is something much more graceful. DNA is not harsh or mechanical. It is soft in form, precise in function, and gently persistent. It is a whisper, a spiral, a thread passed down generation after generation, carrying within it the full instruction set for building a living being. At first glance, DNA may seem impossibly simple. It is made of just a few repeating parts, and yet, from these parts emerges the entire variety of life, from the hum of bees to the stillness of a sleeping bear, from ocean plankton to human laughter. All are shaped by this quiet molecule. To understand DNA, we begin with its shape, the iconic double helix. You might imagine it as a twisted ladder, spiraling gently through space. Each side of the ladder is a backbone, made from alternating sugar and phosphate groups. These run in opposite directions, like two one-way paths winding beside one another. Between them are the rungs pairs of molecules called nitrogenous bases. It is in the arrangement of these bases that DNA stores its messages. There are 4 kinds of bases: adenine, A, thymine, T, cytosine, C, and guanine, G. Each base has a precise partner. Adenine always pairs with thymine, and cytosine always pairs with guanine. This consistency forms the stable core of the molecule. These base pairs are held together by hydrogen bonds, not strong bonds, but gentle ones, just enough to keep them in place, yet soft enough to be opened when needed. When cells need to copy their DNA or read its instructions, these bonds can be unzipped, like a slowly opening book. One strand becomes a template, guiding the formation of a new complementary strand. In this way, the information is duplicated with astonishing accuracy. Each base tells the cell what its partner should be, and the spiral reforms unchanged, preserved. But what does DNA actually say? The answer is proteins. DNA is not a blueprint for your entire body, but for the components your body builds and uses. It tells the cell how to make specific proteins, long chains of amino acids, folded into intricate shapes, each with a particular task. Some proteins carry oxygen, others build muscle, still others speed up chemical reactions, or form structures like hair and skin. To produce these proteins, the cell must first read the gene, a section of DNA that contains the instructions for a specific protein. This reading begins with transcription, a process in which a segment of DNA is copied into a related molecule called RNA. RNA is like a temporary note, made to leave the quiet sanctuary of the nucleus and carry the message into the active space of the cell. Once in the cytoplasm, the RNA finds a ribosome, which reads its sequence 3 letters at a time. Each trio of letters, called a codon, corresponds to one amino acid. In this way, the RNA directs the ribosome to link amino acids in the correct order, forming a protein that the cell needs. This process, called translation, is one of life's most fundamental rhythms, a seamless exchange between instruction and action, information and form. And all of this begins with the careful sequence of base pairs in DNA, A with T, C with G, repeated thousands, even millions of times. The average human gene is about 27, 0 bases long. The entire human genome contains over 3 billion such letters. A single mistake, the wrong base in the wrong place, can sometimes lead to profound consequences, while in other cases, it may have no effect at all. These small variations, called mutations, are the source of genetic diversity. They occur naturally, over time, or through exposure to certain influences. Most are harmless. Some are beneficial. A few may lead to inherited conditions. But taken together, they are the reason no two people, not even identical twins, are truly identical. They are the raw material of evolution, change, and possibility. And yet, for all its complexity, DNA does not act alone. It is influenced by the cell's needs, the organism's stage of life, and even the environment. Some genes are active only during development, others remain dormant until triggered. The way DNA is packaged, the presence of chemical markers, the folding of chromatin, all of these factors determine what is read and when. In this way, DNA is not just a script, but a living manuscript, one that can be annotated, edited, and partially silenced. This fluidity is part of what makes genetics so fascinating and so peaceful in its unfolding. It is not rigid, it flows. As you drift deeper into rest, imagine holding a long ribbon of DNA in your hand. It is soft and light, coiled like ivy around your fingers. As you unroll it, base by base, you begin to see the code, an endless series of letters, forming words your body already knows how to read. No translation is needed, no sound, no effort, just the quiet understanding that this code is already within you, being read, interpreted, and followed, all while you sleep. The double helix itself is not alive, but it gives rise to life. It does not breathe, yet it builds lungs. It does not think, yet it carries the code for a brain. It is, in the truest sense, a quiet architect, always present, always working, always whispering. DNA is where the story of heredity becomes real, where memory becomes molecule, where biology becomes poetry. 6 genes. Sentences in the story of life. As you continue to rest, let us now shift our focus from the full spiral of DNA to its meaningful fragments, those small, quiet sections that carry precise instructions, like sentences written in an ancient language. These are genes, and they are the storytellers within the genome. A gene is not a thing you can hold or see with the naked eye. It is not a separate entity from DNA, but rather a segment, a stretch of the spiral that contains a meaningful instruction. Each gene occupies a particular place on a chromosome, and each one encodes the recipe for a specific protein, or in some cases, a regulatory function that influences other genes. You might think of a gene as a sentence within a very long manuscript. The manuscript is your DNA. The sentence, the gene, makes sense only when the letters are in the correct order. It begins at a specific point, continues through a series of carefully arranged words, and ends in a signal that says, stop. If read properly, it results in something functional, a protein, an enzyme, a silent signal within a cell. The average person carries about 20, 0 to 25, 0 genes. These genes, together, make up what is called the genome, your complete set of genetic instructions. Every cell in your body, except for a few specialized types like red blood cells, carries this entire set, and yet, each cell reads only the pages it needs, only the genes that are relevant to its role. In this way, the genome is not a single story being read aloud. It is more like a vast collection of scripts, each one waiting quietly, to be read only when the time is right. Let's imagine a single gene. It might be the gene that encodes melanin, the pigment that gives color to your skin, hair, and eyes. Inside the DNA, the sequence of bases A, T, C, and G is arranged in a very specific order. This order is the code that tells the cell how to build the protein enzymes responsible for producing melanin. When the gene is needed, it is first transcribed into RNA. Then, that RNA leaves the nucleus and heads to a ribosome, where it is translated into a chain of amino acids. These amino acids fold into a working enzyme that then carries out its role, quietly influencing how much pigment your cells produce. And all of this begins with a single gene, a short, silent section of DNA, not even visible under a microscope, yet powerful enough to shape something as visible as eye color or as vital as metabolism. Genes do not work in isolation. They are part of larger networks, families of genes that interact, support, and sometimes regulate each other. Some genes are structural, helping to build the body's components. Others are regulatory, turning other genes on or off. There are even non-coding regions once called junk DNA that scientists now believe play subtle roles in controlling when and how genes are expressed. And while most genes are shared among all humans, small variations exist between individuals. These variations are called alleles. An allele is simply a different version of a gene. You might carry one allele for curly hair, while another person carries one for straight. You might have an allele that makes your muscles slightly more efficient at using oxygen, or one that gives your earlobes a certain shape. These variations are inherited from your parents, one allele from your mother, one from your father, forming pairs that determine how traits are expressed. Sometimes the two alleles are the same, sometimes they differ. In cases where one is dominant and the other recessive, the dominant allele usually determines the trait that appears. Other traits are polygenic, meaning they are influenced by many genes working together. Height, skin color, and intelligence are examples of complex traits shaped by dozens, even hundreds of genes, each one contributing a tiny piece of the final picture. Genes can also be influenced by environmental factors. A gene might carry the potential for tall stature, but without proper nutrition during childhood, that potential may never fully develop. This gentle interplay between genetics and environment is one of biology's most fascinating harmonies. a soft duet between nature and nurture. Sometimes, the story told by a gene is interrupted. A single letter may be missing, or a small sequence might be repeated too many times. These changes, called mutations, can sometimes lead to genetic conditions. And yet, not all mutations are harmful. Some are neutral. Others provide advantages, leading to natural variation across populations. Even within the same person, genes behave differently across time. During development, some genes are active only in the womb. Others awaken during puberty. Some may stay silent until the body responds to an injury or a shift in the environment. The genome is not static. It is a quiet, adaptive system, listening, responding, and adjusting as needed. It's worth noting that not all species have the same genes. Some share many humans and chimpanzees, for instance, share about 98–99% of their DNA. Yet even small differences can have significant effects. A few changed letters here and there, a different arrangement, a different timing in when a gene is turned on. These subtleties create the rich variety of life we see around us. And so, as you settle further into stillness, imagine your genome not as a machine, but as a library. Each gene is a volume on the shelf. Some are thick, some thin, some are read every day. Others gather dust until a moment of need. The cell knows which to choose. It reads carefully. It follows the instructions, and it builds the quiet machinery of life, one word, one sentence, one protein at a time. Each gene is a sentence in your biological story, a sentence that began long before you were born, a sentence that continues to be read softly, precisely even now, as you drift into sleep. 7 chromosomes, carefully folded threads. As you breathe slowly in the comfort of night, your body enters a deeper stillness. And within that stillness, in every one of your cells, something remarkable remains at rest, yet always ready. These are chromosomes, the carefully folded threads that house your genetic code, stored with such precision that an entire lifetime of biological instructions can fit inside a single cell. Let us imagine again the interior of the nucleus, the small, dense center of your cells, where your DNA quietly resides. The DNA molecule, when fully stretched out, would be about 2 meters long in every cell, and yet it fits neatly, compactly, gently coiled within a space far smaller than the width of a hair. This incredible organization is made possible through the elegant folding of DNA into structures known as chromosomes. A chromosome is not something added to the DNA. It is the DNA itself tightly wound, looped, and packaged with the help of proteins called histones. These histones act like tiny spools around which the DNA is wrapped. The wrapping is not rough or careless. It is intricate, layered, and secure, ensuring the DNA remains both compact and accessible when needed. The combination of DNA and these supporting proteins forms a material called chromatin. When the cell is at rest, not dividing, the chromatin is loosely arranged, allowing the cell's machinery to read from it freely. But when the time comes for a cell to divide, the chromatin condenses further, drawing itself into the familiar X-shaped structures we know as chromosomes. In humans, there are 23 pairs of chromosomes, for a total of 46 in most body cells. One set of 23 comes from your biological mother. The other set comes from your biological father. Each pair contains the same kinds of genes, arranged in the same order, though the versions, or alleles, may differ. This pairing ensures that you carry 2 copies of each gene, providing a gentle balance and a layer of redundancy in your biology. The chromosomes are numbered from 1 to 22, arranged by size, with chromosome 1 being the largest. These are called autosomes, and they carry the vast majority of your genetic material. The 23rd pair are the sex chromosomes, which determine biological sex and related traits. If you have 2 X chromosomes, your biological sex is typically female. If you have 1 X and 1 Y, it is typically male. These chromosomes carry not only sex-determining genes, but many others that influence development and health. Each chromosome contains hundreds to thousands of genes, spaced out along its length like neighborhoods on a long, winding street. Between the genes lie stretches of non-coding DNA, once considered unimportant, but now thought to play roles in gene regulation, structure, and timing. The folding of DNA into chromosomes is more than just clever packaging. It is essential for cell division, especially the process called mitosis, in which one cell becomes two. During mitosis, each chromosome is duplicated, forming two identical strands called sister chromatids, connected at a point called the centromere. These chromatids are then pulled apart into the two new cells, ensuring that each receives a full, identical set of chromosomes. There is also a special form of division meiosis used only for the creation of egg and sperm cells. In meiosis, the number of chromosomes is halved. Each resulting sex cell receives just 23 chromosomes, one from each pair. When an egg and sperm unite during fertilization, the full set of 46 is restored, and a new, unique combination of genetic material is formed. Occasionally, an error occurs during this process. A chromosome may be missing, duplicated, or altered. Some of these changes are harmless. Others may lead to genetic conditions such as Down syndrome, which is caused by having 3 copies of chromosome 21. These variations remind us of the delicate precision required to transmit life's instructions across generations, and the quiet strength of cells that carry out this task millions of times each day. Chromosomes are not static. They do not simply exist in storage. They are interactive, responsive, and dynamic. Genes can be turned on or off depending on signals from the cell or the environment. The folding of chromatin can tighten to silence a gene, or loosen to allow expression. In this way, the shape and structure of chromosomes are part of the message, not just the content. Even the ends of chromosomes hold special significance. These are called telomeres, repetitive sequences that act like protective caps. Each time a cell divides, its telomeres shorten slightly, like the slow ticking of a biological clock. Over time, as telomeres become too short, the cell may stop dividing, entering a state of quiet rest, known as senescence. Telomeres are a focus of research into aging, health, and longevity, a reminder that even at the molecular level, time leaves its trace. As you rest, picture these chromosomes not as hard, geometric shapes, but as delicate threads, glowing softly within the nucleus. Imagine the gentle coil of DNA around histones, the slow condensation as a cell prepares to divide, the careful unfolding as a gene is needed and read. Each chromosome is a library shelf, carefully organized, each volume folded just so, protected and preserved. They do not speak, yet they carry stories. They do not move quickly, yet they travel through time from generation to generation, cell to cell, being duplicated, shared, and remembered. In their quiet structure, we find the blueprint of life not merely stored, but alive, changing, evolving, and responding. And as you drift deeper into sleep, you carry them within you, all 46, resting gently in your trillions of cells, continuing the silent sacred work of heredity, night after night, moment after moment, in perfect, peaceful rhythm. 8. The genetic code, life's quiet alphabet. Somewhere deep within you, in the gentle stillness of each cell, a language is spoken not in sounds, but in symbols, not in ink, but in molecules. It is a language older than words, older than memory, a language so elegant and quiet, it can guide the growth of forests, the formation of feathers, and the beating of your very own heart. This is the genetic code, and it is the alphabet of life. At its heart, the genetic code is incredibly simple, made from just 4 letters. These letters are the nitrogenous bases we met earlier in DNA. Adenine A, thymine T, cytosine, C, and guanine, G. These are the characters in the molecular script, and when arranged in long sequences along the DNA strand, they create the instructions for building and operating a living organism. The elegance lies in the order, not in the bases themselves, but in how they are arranged. The cell reads these letters 3 at a time, forming what are known as codons. Each codon acts like a word in this soft language, and each one corresponds to a specific amino acid, the building blocks of proteins. There are 64 possible codons, but only 20 amino acids. This means the code has some flexibility. Some amino acids are represented by more than one codon. This built-in redundancy helps protect against small errors, allowing the system to function smoothly, even when a letter occasionally shifts. For example, the codon AUG always signals the beginning of a protein. It codes for the amino acid methionine, and it tells the cell, begin here. From there, the cell continues reading codons, each telling it which amino acid to add next to the growing protein chain. Eventually, it encounters one of 3 stop codons, signaling that the protein is complete. This quiet reading takes place during the process known as translation, where the message carried by messenger RNA, mRNA, is read by a ribosome, one codon at a time. Each codon is matched with a corresponding transfer RNA, tRNA molecule, which carries the appropriate amino acid. The ribosome then links these amino acids together in sequence, forming a protein, a soft ribbon of function, folded with care. All of this unfolds with incredible speed and precision, and yet it is silent. There are no shouts of command, no visible movements, just a steady procession of letters and molecules, gently cooperating to build the tools that sustain life. What makes the genetic code so powerful is its universality. With very few exceptions, all living organisms, from the tiniest bacteria to the tallest trees, use the same genetic code. The same codons mean the same amino acids. Whether in a frog, a flower, or a person, this shared code is one of the strongest pieces of evidence that all life on Earth is connected, descending from a common origin. It is as if life chose a single alphabet long ago and never changed it, a choice so quiet, so unassuming, that it carried on unchanged for billions of years. The fact that this code is both simple and consistent allows scientists to do remarkable things. Genes from one organism can be introduced into another and still be understood. A bacterial cell can be given a gene from a human and still produce the corresponding protein, so long as the code is written correctly. This forms the basis of genetic engineering, where careful editing of DNA can lead to helpful changes in medicine, agriculture, and research. Yet, despite all this scientific potential, the code itself remains humble. It is not poetic in the way we think of words. It has no style, no emotion, and yet, in its simplicity, it forms the basis of all biology. It determines eye color, blood type, metabolic rate. It tells a cell when to grow and when to rest, what to build and what to release. Sometimes, small changes in the sequence, a mutation, can alter a codon. Depending on where the change occurs, it may have no effect, or it may lead to a different amino acid being used. Occasionally, it introduces a stop codon too early, halting the protein before it's complete. These gentle shifts in the code can influence traits, health, or adaptation, adding to the quiet variability of life. The genetic code is also directional. DNA is read in one direction, from a starting point to a stop. This ensures that each gene is interpreted consistently. It is not enough to have the right letters, they must also be in the right order. Read from the right place, spaced just so, like a sentence in a book. Even a single letter out of place can change the meaning. Despite this complexity, the cell reads the code effortlessly. It never questions, never hesitates. From birth to death, in trillions of cells, the genetic code is read and interpreted unceasingly. The machinery that performs this work is ancient and automatic, a gentle, molecular ritual passed down since the first spark of life flickered on Earth. As you rest tonight, you are not aware of the codons being read within you. You do not feel the proteins being built, but they are there silently forming, folding, and functioning in the darkness. Every cell in your skin, every fiber in your muscles, every corner of your brain, all of them built and maintained by this quiet alphabet. You are, in many ways, a story told in A, T, C, and G. And as the night deepens and your thoughts grow still, imagine this code stretching out like a ribbon of light, looping gently within every cell, repeating the same ancient song. It is not loud. It does not rush. It does not falter. It simply continues, letter by letter, codon by codon, forming you, sustaining you, softly and silently. 9 RNA, the messenger between worlds. As your thoughts soften and your body becomes heavier with sleep, we return once more to the calm interior of the cell. Here, where silence reigns and every process unfolds with grace, there is a bridge, a tender link between the stillness of the nucleus and the movement of the wider cell. That link is called RNA, or ribonucleic acid. While DNA remains safely stored in the nucleus, the central archive of life's information, RNA acts as its gentle courier. It listens, it copies, it travels, and in doing so, it brings the message of DNA out into the cell, where it can be interpreted and transformed into the machinery of life. In essence, RNA is the messenger between worlds. It is the voice of the genome, carried softly from storage to action. The structure of RNA is similar to DNA, but with a few key differences. Like DNA, RNA is composed of nucleotides, each containing a sugar, a phosphate, and a nitrogenous base. However, RNA uses the sugar ribose, while DNA uses deoxyribose, hence their names. And in RNA, the base uracil takes the place of DNA's thymine. So the pairings change slightly. Adenine now matches with uracil instead of thymine. Another gentle distinction. DNA is a double-stranded helix, a twisted ladder, while RNA is single-stranded, a flexible ribbon, able to fold, loop, and drift with ease. This lighter structure allows RNA to leave the nucleus, moving freely through the cytoplasm. There are many types of RNA, each with its own soft role, but the one we most often speak of is messenger RNA, or mRNA. This is the form that carries the genetic message from DNA to the ribosome, the site where proteins are built. The journey begins with a process called transcription. Imagine the DNA unwinding gently, exposing a single gene, a short stretch of bases with instructions for one particular protein. As the DNA opens, an enzyme called RNA polymerase begins to read the sequence. It moves slowly along the template strand, matching each DNA base with its RNA counterpart, A with U, T with A, C with G, and G with C. Base by base, a strand of RNA forms a soft mirror of the gene it has copied. This mRNA strand carries the exact instructions needed to assemble a specific protein. Once the message is complete, the RNA strand detaches, the DNA coils quietly back into place, and the mRNA prepares to leave the nucleus. This moment is a gentle transition. The message, once sealed away in the quiet heart of the cell, is now ready to travel. The mRNA exits the nucleus through nuclear pores, tiny openings in the nuclear membrane, and enters the cytoplasm, where it floats toward a ribosome. The ribosome, like a patient reader, binds to the mRNA and begins to interpret its message. This is the start of translation, where RNA's code is converted into a chain of amino acids, the beginnings of a protein. But mRNA is not the only type of RNA involved in this process. There is also transfer RNA, or tRNA, small, folded molecules that carry amino acids to the ribosome. Each tRNA recognizes a specific codon on the mRNA and delivers the correct amino acid to match. One by one, these amino acids are linked together, forming a protein chain that folds and curls into a precise, functional shape. Another quiet helper is ribosomal RNA, or rRNA, which forms the structural core of the ribosome itself. It ensures the process of translation unfolds smoothly, holding the mRNA in place and coordinating the delivery of each new building block. Together, mRNA, tRNA, and rRNA create a team of translators, turning the genetic code into living function. Each does its part without hurry. Each follows the same timeless pattern, day after day, cell after cell. Unlike DNA, which is preserved and protected, RNA is temporary. It exists only as long as needed a single message for a single task. When the protein has been built, the mRNA is quietly broken down by enzymes and recycled into new materials. In this way, the cell keeps the process efficient, responsive, and ever adaptable. RNA also plays many additional roles beyond protein synthesis. There are small interfering RNAs that help regulate gene expression, silencing specific messages when they're no longer needed. There are microRNAs that fine-tune protein production, adjusting levels with gentle precision. And in the simplest organisms, like some viruses, RNA is used as the genetic material itself, replacing DNA entirely. These variations show RNA's flexibility. It is not rigid or fixed. It flows, it folds, it adapts. In a way, it is the breath of the cell, the quiet exhale of information, moving from storage into motion. And though it is fleeting, RNA is essential. Without it, DNA would remain silent. The genes, so carefully stored in chromosomes, would never be expressed. The proteins, so vital to every cell, would never be built. As you sleep, this entire process continues within you, in your skin, in your muscles, in your blood. Genes are being transcribed, messages are being carried, proteins are being made. Every moment, in every tissue, RNA walks the path from code to function, message to meaning. It asks for nothing. It leaves no trace. It simply delivers. So as your breath deepens and you drift into the soft currents of sleep, imagine the quiet path of a single mRNA strand, floating gently from the nucleus, reaching its ribosome, being read codon by codon, word by word, and then, fading back into the cell, its message fulfilled. RNA is not a keeper of memory. It is the voice of memory, a soft, momentary whisper, a messenger between the stillness of instruction and the movement of life. 10 proteins, builders, movers, and messengers. Now that we've followed the quiet message of RNA as it carries life's instructions from the depths of the nucleus to the open spaces of the cell, we turn now to the products of that message, the final form, the instruments of action. These are the proteins, delicate chains of function that do nearly everything life requires. If DNA is the library and RNA is the messenger, then proteins are the builders, movers, and messengers of the cell. They are the ones that take what is written and bring it to life. Let us begin at their origin. A protein starts as a long, single chain of smaller units called amino acids. Each amino acid is like a letter in a new sentence, and the sequence in which they are arranged determines the protein's final shape and its role. There are 20 standard amino acids, and while this number is small, the combinations are nearly infinite. Much like words in a language, the meaning changes completely depending on the order. A chain of 100 amino acids could be arranged in countless ways, each forming a unique protein with a unique shape and a unique task. After the ribosome reads the mRNA and assembles the amino acid chain, the new protein begins to fold. This folding is not random. It is guided by the chemical nature of each amino acid, how they interact with one another, how they attract or repel, and how they seek water or avoid it. The result is a three-dimensional structure, often with multiple levels of folding: primary, secondary, tertiary, and in some cases, quaternary. The shape of a protein is everything. It determines what the protein can do, where it can go, and how it will interact with other molecules. Even a small mistake in folding caused by a single incorrect amino acid can render a protein ineffective or even harmful. But when built correctly, proteins become the gentle machinery of life. Some proteins are structural, providing strength and support. One familiar example is collagen, found in skin, tendons, and connective tissues. Another is keratin, which forms your hair and nails. These proteins give the body form and resilience, not with hardness, but with silent strength. Other proteins are enzymes, which speed up chemical reactions that would otherwise occur far too slowly to support life. Without enzymes, the body could not digest food, copy DNA, or produce energy. One enzyme, amylase, begins breaking down starches in your mouth as you eat. Another, DNA polymerase, helps replicate your genetic material before each cell divides. Then there are the transport proteins, which carry substances through the bloodstream or across cell membranes. One of the most well-known is hemoglobin, the protein in red blood cells that binds to oxygen and carries it from your lungs to every corner of your body. Without hemoglobin, your cells would not receive the oxygen they need to survive. Proteins can also act as messengers, especially in the form of hormones. These signaling proteins travel through the body, delivering quiet instructions to distant cells. Insulin, for example, helps regulate blood sugar by telling cells when to absorb glucose. Growth hormone influences cell development, especially in childhood, and oxytocin, sometimes called the bonding hormone, plays a role in emotional connection and trust. Still more proteins form the immune system, acting as guards and scouts, identifying intruders and helping neutralize infections. Antibodies built by white blood cells recognize specific shapes on viruses and bacteria. Each antibody is tailored to its target, marking it for destruction or disabling it directly. This precision allows your body to protect itself with subtlety and efficiency, all orchestrated by protein molecules. And there are proteins inside your cells that help them move. These are the motor proteins, which travel along cellular highways, carrying cargo from one part of the cell to another. One such protein, kinesin, looks almost like it is walking a tiny, step-by-step journey along molecular tracks, carrying vesicles full of materials to where they are needed. In every corner of your body, proteins are working not loudly, not with drama, but with constancy. They don't think, they don't choose, but they perform with grace and repetition, doing what their structure allows and requires. Every protein is coded by a gene, translated from a sequence of DNA through the gentle process we have already explored. But after it is made, many proteins are modified, folded further, trimmed, or combined with other molecules. These subtle touches refine their shape and purpose, adapting each protein to the specific needs of the cell and the moment. Proteins also have lifespans. Some last only minutes. Others, like those in eye lenses or cartilage, may last years. When they are no longer needed or when they become damaged, proteins are marked for removal. Specialized enzymes quietly break them down into amino acids, which are then reused to build new proteins. In this way, the cycle continues. Build, serve, break down, rebuild. Even as you lie in stillness, your body is filled with these silent workers. Your muscles rest, but the proteins that power them remain. Your mind slows, but the proteins that regulate thought and emotion continue their work. In every cell, proteins are folding and unfolding, guiding the balance of life. Imagine now a cell at peace, a space of quiet activity. Tiny ribosomes assemble amino acid chains, Proteins drift into the endoplasmic reticulum, folding in gentle pockets of the membrane. Enzymes brush past one another, working without pause. The air is still, but the machinery never stops. No noise, no rush, just life, carried out molecule by molecule, moment by moment. Proteins are not permanent. They do not last forever. But while they are here, they act with a quiet purpose, born from the code of DNA, delivered by RNA, and shaped by chemistry. They do not command attention, but they build, they move, they heal. And so, as you drift into deeper sleep, let the image of these proteins stay with you, not as sharp machines, but as ribbons and spirals, soft and folded, carried gently within every cell, working not for praise, but for peace. 11 cell division, passing the blueprint on. As you drift deeper into rest, your body, though still and quiet, continues a soft rhythm, one of replication, renewal, and continuity. This rhythm plays out not in your conscious awareness, but in your cells. It is the rhythm of cell division, where life's blueprint is gently copied, packaged, and passed forward. Cell division is the process by which cells reproduce. It is how your body grows, how it heals, and how it maintains itself throughout your life. Even now, as you sleep, millions of your cells are in various stages of division, carefully ensuring that each new cell receives an identical set of genetic instructions. There are two primary forms of cell division: mitosis and meiosis. Each has its own purpose. Each unfolds with a calm precision. Let us begin with mitosis, the process used for growth, repair, and everyday cell renewal. In mitosis, one cell divides to produce two genetically identical daughter cells. Each one receives a complete copy of the original DNA, every gene, every chromosome, every letter. This ensures that your skin cells remain skin cells, your liver cells remain liver cells, and that each tissue maintains its identity and function. The journey of mitosis begins with a preparatory phase known as interphase. During this time, the cell is not dividing, but it is active, growing, performing its normal duties, and most importantly, replicating its DNA. Each chromosome is copied, forming two identical strands known as sister chromatids, joined together at a point called the centromere. Once the DNA is fully duplicated, mitosis gently begins. It unfolds in a series of stages, each one moving the process forward with quiet grace. The first stage is prophase. The chromatin, that soft, loose arrangement of DNA, begins to condense into distinct chromosomes. The nuclear membrane slowly dissolves, and structures called spindle fibers begin to form from the centrosomes, drifting to opposite ends of the cell. Next comes metaphase, a moment of balance. The chromosomes line up along the center of the cell, forming a straight, even line. Each chromosome is attached to spindle fibers that stretch out from either side, ensuring a perfect tension, a symmetry that prepares the cell for its final step. Then comes anaphase, the gentle parting. The sister chromatids are pulled apart, drawn to opposite poles of the cell. The identical copies of the DNA move away from one another, like the opening of two arms, ensuring that each new cell will receive the full set. Finally, in telophase, the cell prepares for completion. The chromosomes begin to unwind, returning to their softer chromatin state. New nuclear membranes form around each set of DNA, and the cell begins to divide its cytoplasm. This final split, called cytokinesis, completes the process. The cell gently pinches in two, forming two new daughter cells, each a mirror of the original, each ready to begin its own quiet cycle. Mitosis happens continuously throughout your body, in your skin, where old cells are shed and replaced, in your bone marrow, where blood cells are renewed, in your gut lining, where cells live brief but essential lives. Each time, The process is carried out with delicate care, a quiet commitment to continuity. Now let us turn to meiosis, a more specialized and complex form of division, used only to create gametes, sperm, and egg cells. These are the cells involved in reproduction, and their task is to carry half the genetic material, 23 chromosomes, instead of 46, so that when two gametes unite, a new, complete set is formed. Meiosis begins similarly to mitosis, with a round of DNA replication. But instead of one division, meiosis involves two rounds, meiosis I and meiosis II, resulting in 4 daughter cells, each with half the original DNA. During meiosis I, something extraordinary occurs. Homologous chromosomes, one from each parent, pair up and exchange small sections in a process called crossing over. This quiet exchange increases genetic diversity, ensuring that no two gametes are exactly alike. It is a gentle reshuffling of the deck, a soft stirring of inherited traits. The homologous pairs are then separated into 2 cells. Each cell now contains a mixed set of chromosomes, still doubled, but already unique. In meiosis II, these chromosomes are split once more, separating the sister chromatids. The result is 4 haploid cells, each with just one copy of each chromosome ready to participate in the creation of new life. This entire process is deeply conservative and creative at once. It preserves the species, while allowing for endless variation. It maintains the code, while mixing the stories. In every child, this gentle dance of chromosomes brings together traits from generations past, writing new combinations without noise or force. Both mitosis and meiosis are made possible by the same molecules we've come to know. DNA, coiled into chromosomes, RNA, preparing the cell's components, proteins, guiding the division, and enzymes, ensuring that each step is performed without error. Though it is a physical process, cell division is also a kind of memory, a quiet remembering of form, function, and identity. It is how a fertilized egg becomes an infant, and how that infant grows. It is how a cut heals, a bone mends, a thought becomes memory through new neural connections. It is how the body knows how to continue, without being told. Even now, cells are dividing softly within you, not in chaos, but in calm, a trillion little acts of copying, arranging, parting, and settling into new places. Each new cell carries the story of the last, and each does its part in the long, uninterrupted continuity of life. So as you lie still, perhaps imagine this soft unfolding within, a nucleus opening like a flower, DNA drawing apart, and new cells gently forming invisible, soundless, perfect in their timing. In cell division, we see that life is not a single act, but a continuous unfolding, an unbroken passing of the blueprint from one cell to another, from one generation to the next, and always with quiet care. 12. Inheritance. Patterns through generations. By now, we have followed the thread of life from the spiral of DNA, through the whisper of RNA, to the work of proteins, and the peaceful rhythm of cell division. All of these processes prepare us for one of life's most enduring wonders, inheritance. The quiet transfer of traits, through time, from one generation to the next. Inheritance is the reason why children resemble their parents, why your eyes may mirror your grandfather's, or your voice carry the lilt of a parent's tone. It is not magic. It is memory, biological memory, stored in genes and gently passed onward. At the heart of inheritance are genes, nestled along the length of chromosomes. Each gene may have different alleles, alternative forms that result in different expressions of a trait. For example, a gene responsible for flower color in a plant might have one allele for red petals and another for white. How these alleles are inherited and which ones are expressed follows a soft logic, first described in the 1800s by a quiet monk named Gregor Mendel. Tending his garden of pea plants, Mendel noticed patterns in how traits appeared across generations. Round peas or wrinkled, tall stems or short. These observations led to the basic principles of inheritance, often called Mendelian genetics. Mendel's first key idea was the concept of dominant and recessive alleles. If an organism inherits two different alleles for a gene, one from each parent, the dominant allele is the one that shows itself in the physical trait, or phenotype. The recessive allele remains silent, its effects hidden unless it is paired with another copy of itself. For example, if a plant inherits one allele for purple flowers, dominant, and one for white, recessive, it will bloom in purple. Only when two recessive alleles are present, one from each parent, will the plant show the white flowers. This leads to the next principle, the idea of genotype versus phenotype. The genotype is the genetic makeup, the alleles present in the DNA. The phenotype is the observable trait, the result of how those alleles are expressed. Two individuals might look the same outwardly, sharing the same phenotype, while carrying different genotypes beneath the surface. Another principle Mendel discovered was the law of segregation, the idea that during the formation of gametes, the two alleles for each gene separate. Each gamete receives only one allele from each pair. This ensures that when fertilization occurs, the offspring receives one allele from each parent, restoring the full set of genetic instructions. A further principle is the law of independent assortment, the understanding that genes for different traits are inherited independently of one another, provided they are located on different chromosomes. This allows for new combinations of traits in each generation, a quiet mixing that increases variety and uniqueness. Though Mendel's work focused on simple traits with clear patterns, modern genetics has shown that many traits are more complex. These are called polygenic traits, meaning they are influenced by multiple genes working together. Examples include height, skin color, eye color, and even susceptibility to certain health conditions. In these traits, inheritance is not a single yes or no, but a spectrum shaped by the combination of many alleles, each adding a small effect. This makes inheritance both predictable and unpredictable, structured and full of surprises. Some traits are also influenced by incomplete dominance, where neither allele is completely dominant. In such cases, the phenotype is a blend. For example, a red-flowered plant crossed with a white-flowered one might produce pink offspring, a soft merging of both. In other cases, codominance occurs, where both alleles are expressed equally. A classic example is blood type. A person with one A allele and one B allele has AB blood, expressing both traits without blending them. Then there are sex-linked traits, which are carried on the sex chromosomes, particularly the X chromosome. Since males have only one X chromosome, any allele carried there, even if recessive, is more likely to be expressed. This is why certain traits, like red-green color blindness or hemophilia, are more common in males. As generations pass, these inherited traits weave together to create the vast diversity of life, the gentle variety seen in faces, fur patterns, feather hues, and leaf shapes. Each individual carries not just traits from their parents, but echoes from distant ancestors, stretching back through time, passed from cell to cell, quietly remembered. Sometimes, traits skip generations. Sometimes they appear unexpectedly. These surprises do not defy genetics, they are part of its subtlety. A recessive allele may be carried silently for years, hidden in the code, until the right combination brings it to the surface. And though we often think of inheritance as fixed, you have your genes, and that's that there is growing understanding of epigenetics, the gentle influences that can alter gene expression without changing the DNA itself. Diet, environment, stress, and exposure can all leave soft marks on the genome, turning genes on or off like a dimmer switch. Some of these changes may even be passed to offspring, forming a second layer of inheritance, one shaped not by sequence, but by experience. This quiet, layered transmission of genes, of traits, of patterns, forms the biological lineage that links every living being. It is a thread that stretches through time, connecting you not just to your parents, but to your grandparents, and theirs, and all who came before. Even now, as you sleep, your cells hold these inherited patterns. Every freckle, every curl of hair, every rhythm of your heartbeat carries the influence of those who lived before you. And when cells divide, or when new life begins, those patterns continue reshaped slightly, softly, but always remembered. Inheritance is not just a science. It is a story, written in molecules, passed hand to hand through generations, and it continues still quietly, faithfully, beautifully. 13. Genetic variation. The gentle differences between us. As night deepens and the mind grows still, we turn now to one of the softest truths of biology, that no two living beings are exactly alike. Even among siblings, or twins, or petals from the same stem, subtle differences exist. These variations are not mistakes, nor flaws. They are the quiet brushstrokes that nature uses to paint individuality. This gentle uniqueness begins with genetic variation, the natural differences in DNA sequences from one organism to another. It is the reason for the endless variety we see in eye color, facial features, voice tone, metabolism, and more. It is why fingerprints are never repeated, and why even your own cells may carry slight variations as they age. At the heart of this variation are alleles, the different forms of a gene. Most genes have at least two alleles, though some have many more. The combination of alleles you inherit from your parents, along with how they are expressed, contributes to your distinct traits. Even small changes, a single base swapped for another, can gently alter the outcome. These changes are called mutations. Though the word might sound harsh, in biology, mutations are simply natural changes in the DNA sequence. Some are inherited from parents. Others occur spontaneously during DNA replication. Still others arise from environmental influences, sunlight, radiation, or even the byproducts of your own metabolism. Most mutations are neutral. They neither help nor harm. They simply exist as part of the code. Some may have beneficial effects, offering protection against certain diseases or improving efficiency in a cellular process. Others may increase susceptibility to health conditions, though rarely in isolation. More often, it is the gentle interplay of multiple genes, along with environmental factors, that shapes such outcomes. Another important source of variation comes from sexual reproduction. Each time sperm and egg unite, they bring together two sets of DNA, combining them in a unique way. Thanks to independent assortment, crossing over, and random fertilization, each offspring receives a distinct mix of genes, not a perfect half and half, but a blend influenced by chance and recombination. Let us briefly revisit crossing over, a soft shuffle that occurs during meiosis. As chromosomes pair up, they sometimes exchange small sections with each other. These tiny swaps create new combinations of alleles on a single chromosome, combinations that may never have existed before. The result is greater diversity, even among siblings from the same parents. There is also variation introduced by the presence of gene duplications, insertions, or deletions, where sections of DNA are added, repeated, or removed. Some of these changes have no visible effect, while others may influence traits in more noticeable ways. Over many generations, these small changes accumulate, contributing to the beautiful complexity of life on Earth. Genetic variation exists not only between individuals, but also between populations. Over time, groups of organisms living in different environments may develop distinct genetic profiles. These differences reflect local adaptations traits that helped ancestors survive and thrive in particular climates, diets, or conditions. For example, variations in genes related to skin pigmentation evolved in response to sunlight exposure. Populations near the equator, where sunlight is intense, tend to have darker skin rich in melanin, which provides protection from ultraviolet rays. In regions with less sunlight, lighter skin allows for more efficient synthesis of vitamin D. These variations are not better or worse. They are simply appropriate for the environments in which they arose. Other examples include lactose tolerance, which is common in populations with a long history of dairy farming, and altitude adaptation, seen in peoples living in high mountain regions. In each case, the variation reflects a gentle accommodation, the quiet shaping of biology by geography and culture. Importantly, the vast majority of human genetic variation is shared. Despite our external differences, all humans are over 99.9% genetically identical. That tiny fraction of difference, less than 0.1%, accounts for all visible diversity in height, hair texture, eye color, and beyond. It is enough to make each person unique, and yet also a reminder of our deep biological unity. Genetic variation is not exclusive to humans. Every species, from trees to turtles, fish to fungi, carries its own rich store of variation. It is the raw material of evolution, giving natural selection something to act upon. Without variation, populations could not adapt to change. With it, life remains flexible, resilient, and creative. Some organisms reproduce asexually, producing clones of themselves. Yet even they are not entirely free from variation. Random mutations still occur. Environmental factors may alter gene expression, and over long spans of time, even a clone can become something new. In modern biology, the study of variation is also helping us understand and address human health in more personalized ways. The field of genomics explores not just the sequence of DNA, but how variation influences disease risk, drug response, and treatment effectiveness. This is the basis for personalized medicine care that honors the individuality of each person's genetic profile. And yet, for all its complexity, genetic variation remains a quiet process unfolding invisibly, passed down in the code, expressed softly in the curve of a smile, the shape of a hand, or the rhythm of a heartbeat. You might imagine it now as a garden of possibilities. Each person, a different blossom. Each gene, a seed planted long ago, shaped by winds of ancestry and weathered by time. No two paths are alike, no two expressions the same. And yet, all are part of the same living tapestry. As you sleep, your cells continue to express their unique blend of traits. Some genes are active, others are silent. Some proteins are folded precisely to suit your individual biology. And deep within, your chromosomes carry a pattern of variation that has never existed before and never will again, in exactly the same way. You are not a copy. You are not a repetition. You are a variation, gentle, natural, beautiful. 14 Epigenetics. The quiet marks that shape gene expression. As you drift in stillness, your body settles into its own deep rhythm. Slow breath, steady heartbeat, quiet warmth. Beneath that calm, your cells are not just following inherited instructions. They are also responding, adjusting, and remembering. This gentle ability to change gene expression without altering the underlying DNA is known as epigenetics. The word itself comes from the Greek epi, meaning upon or above, and genetics, meaning the study of genes. Epigenetics, then, refers to a layer on top of genetics, a soft, flexible system that controls how, when, and where genes are used. Imagine your DNA as a vast library of recipes. Each gene is a page in that collection, but in any given cell, only a small fraction of those pages are actively read and followed. The rest remain untouched, folded neatly away. Epigenetics is the mechanism that places bookmarks sticky notes, or gentle folds in the pages, not changing the text itself, but influencing which parts are opened and which are left closed. One of the most common forms of epigenetic modification is DNA methylation. In this process, small chemical groups, called methyl groups, are added to specific bases in the DNA, usually at cytosine residues. These groups act as soft markers, telling the cell, "Do not read this section right now." When a gene becomes heavily methylated, its expression is typically reduced or silenced. The gene remains present in the DNA, unchanged, but its voice grows quiet. Another major form of epigenetic regulation involves the histones, the spool-like proteins around which DNA is wrapped. These histones can be modified by adding or removing chemical tags such as acetyl groups or phosphate groups. These tiny changes affect how tightly the DNA is coiled. When the DNA is loosely wrapped, the cell can easily access and read the genes. When it is tightly coiled, access is limited and the genes remain silent. This shifting of accessibility allows the cell to turn genes on or off as needed, without altering the underlying sequence. In this way, epigenetics acts like a dimmer switch rather than a light switch. Genes are not simply present or absent, they can be turned up, down, paused, or delayed. The result is a subtle, layered regulation of gene activity that allows organisms to respond to their environments with remarkable precision. One of the most beautiful examples of epigenetics in action is cell differentiation. All the cells in your body, skin, muscle, brain, blood, share the same DNA, yet they look and behave completely differently. This is because, during development, certain genes are epigenetically silenced or activated in each type of cell. A liver cell reads only the genes it needs to function as a liver cell. A neuron activates a different subset. Epigenetics ensures that the right parts of the genome are used in the right place, gently guiding each cell toward its identity. These changes can be stable and long-lasting, allowing cells to maintain their roles for a lifetime. But they can also be reversible. Environmental factors, nutrition, stress, toxins, and even experiences can lead to epigenetic changes, some of which may be temporary, while others may last for years. For example, research has shown that diet can influence DNA methylation. Certain nutrients, like folate, vitamin B12, and choline, contribute to methyl group formation. In this way, the foods we eat can subtly shape how genes are expressed, turning up the volume on some messages, turning others down. Similarly, chronic stress has been linked to epigenetic changes in genes related to emotional regulation and immune response. Over time, sustained stress can gently shift how the body responds to challenges, leaving soft marks on the genome without rewriting the code itself. Even more intriguing, some epigenetic modifications appear to be heritable, passed from parent to offspring, along with the DNA. This means that experiences in one generation may influence gene expression in the next, without changing the DNA sequence. In plants and simple animals, this transgenerational epigenetic inheritance is well documented. In humans, the evidence is more subtle, but growing. For example, studies of people born during times of famine suggest that the nutritional experiences of grandparents may influence the metabolism of grandchildren, a soft echo of hardship passed through biology. It is important to note that epigenetics does not override genetics. It works alongside it, a second layer of control, built on top of the inherited code. It cannot create new genes, but it can influence whether and how those genes are used. And because it is responsive, epigenetics offers a bridge between nature and nurture. Your genes provide the structure, the potential, but your environment, experiences, and choices help shape the way that potential is realized. Epigenetics also plays a role in development, aging, and disease. As we grow older, epigenetic patterns change. Some genes that were once silent may become active, others that were active may fade. This shifting contributes to the aging process and to the changing nature of our bodies over time. In certain diseases, especially cancers, epigenetic regulation can become disrupted. Genes that suppress tumor growth may become silenced. Genes that promote unchecked cell division may become overactive. Understanding these patterns has opened new paths for epigenetic therapies, treatments that aim to restore healthy gene expression without altering the underlying DNA. Even now, as you sleep, your cells maintain these patterns. Some genes are active in the quiet hours of rest. Others are gently turned down. The epigenetic marks on your DNA guide this rhythm, instructing the cell when to repair, when to rest, when to restore. You might imagine these marks not as rigid labels, but as soft fingerprints on the surface of your genome, some fresh, some faded, all quietly shaping your biology. There is no sound, no shift you can feel, yet your cells remember, they adjust, they respond. And in this quiet adaptation, we glimpse one of nature's most poetic truths, that biology is not fixed, but fluid, not a static code, but a living, breathing story shaped by time, environment, and experience. As you rest in this moment, your body is already gently preparing for tomorrow, guided by your genes, shaped by your life, carried forward with care. 15 Heredity and Everyday Traits. What we get from our parents. There is something profoundly gentle about resemblance. The subtle way, a smile, a dimple, or the shape of an eyebrow can echo from one generation to the next. As you soften into rest, consider how many of your features, both visible and unseen, carry the quiet fingerprint of your ancestry. This is heredity in its most familiar form, the traits you inherit from your parents. At its heart, heredity is the transmission of genetic information from one generation to the next. With every new life, genes are passed down, half from one parent, half from the other, and in this soft blending, you are formed. Let us begin with the visible traits, those that can be seen at a glance. Eye color, for instance, is one of the most commonly discussed inherited traits. It is determined by the amount and type of melanin in the iris, controlled by several genes. While once thought to follow simple, dominant, and recessive patterns, we now know it is more complex. Many genes contribute, some encouraging the production of melanin for darker eyes, others limiting it for lighter shades. The result is a spectrum: brown, green, hazel, gray, blue, each one a delicate mix of genetic influence. Hair texture and color are similarly inherited. Do you have curls, straight hair, waves? These features arise from the shape of the hair follicle and the distribution of keratin, both shaped by inherited genes. Hair color, too, comes from melanin. For black and brown tones, pheomelanin for reds and blondes. The exact hue depends on which versions of pigment genes you've inherited and how active they are. Then there's height, a trait influenced by hundreds of genes, each contributing a small effect. Together, they form a soft genetic baseline, one that is then modulated by nutrition, sleep, and childhood health. The result is a gentle blend of inheritance and environment, your parents' heights, providing a framework, shaped further by your life's circumstances. Skin tone, too, is inherited not in a simple way, but through a complex interplay of genes that regulate the amount, type, and distribution of melanin. These patterns reflect generations of adaptation to sunlight, geography, and climate, and though we carry them on our skin, they speak quietly of ancestry, of migrations long past, of the soft language of evolution. But not all inherited traits are so visible. Blood type is another example, determined by genes that code for proteins on the surface of red blood cells. The ABO system includes type A, type B, type AB, and type O, depending on which antigens are present. You inherit one allele from each parent, and the combination determines your type. It is a trait silently carried, essential for transfusions, yet invisible in daily life. Taste sensitivity is also influenced by inheritance. Some people carry a version of a gene that makes them more sensitive to bitter flavors, like those found in certain greens or dark chocolate. Others may have inherited a version that softens this response. What you enjoy eating or avoid may trace back quietly to the genes you carry. Even the way your earwax forms dry and crumbly or moist and sticky is shaped by a single gene. This tiny difference may seem unimportant, yet it is a small mark of ancestry. In some regions of the world, one type is more common than the other, forming a subtle genetic map. Your handedness, or preference for using the left or right hand, also appears to be influenced by genetic factors, though not determined by a single gene. It may reflect a combination of inherited tendencies and early developmental cues, forming gently during gestation and infancy. More complex traits like temperament, sleep patterns, and susceptibility to certain health conditions also have hereditary components. While they are shaped by many genes and deeply influenced by life experience, research suggests that some of your inclinations, rhythms, and reactions are softly guided by what you inherit. For instance, your chronotype, whether you're naturally an early riser or a night owl, is partially influenced by genes that regulate your internal clock. Your need for sleep, how deeply you rest, and how easily you wake may all carry a quiet genetic signature. Some inherited traits also relate to sensory perception. The ability to roll your tongue, taste cilantro without it seeming soapy, or hear certain frequencies more easily. These differences often trace back to subtle variations in your DNA. They are not better or worse, just different brushstrokes on the canvas of individuality. And sometimes, traits inherited from parents don't show up immediately or at all. You may carry a recessive allele, silently, without expressing it, but that gene may reappear in a future child or grandchild, carried quietly through the generations until the right pairing brings it forth. It's also worth remembering that heredity is not about replication. You are not a copy of your parents. You are a combination, shaped by two lines of ancestry merging in a moment of chance and wonder. Even siblings with the same parents receive different assortments of alleles, a result of how chromosomes are shuffled and distributed during the formation of gametes. Each trait is a point of connection, a gentle thread that links you to those who came before. And yet, you are something new, a singular arrangement, never before seen, never to be repeated in exactly the same way. As you lie in the quiet of night, consider these connections. The soft echo of your mother's laugh, the thoughtful tilt of your father's brow, the way your hands move, your voice carries, your thoughts form each a reflection, and yet wholly your own. In the long story of heredity, you are a chapter unlike any other written with familiar letters, but arranged into a new, beautiful composition. You carry the past, you embody the present, and through the code you hold, and the choices you make, you quietly shape the future. 16 genetic disorders. When the code shifts, while most of the time, the genetic code flows with quiet harmony, guiding the growth, repair, and rhythms of life, there are times when that code changes in ways that gently shift its meaning. These changes are not always harmful. Many are harmless, or even beneficial, but some lead to genetic disorders, conditions where small alterations in DNA affect the body's function, sometimes from birth, sometimes later in life. Tonight, we'll explore this topic not with worry or fear, but with calm understanding to see how life's instructions, when gently rewritten, may lead to differences that are inherited, managed, and understood with care. A genetic disorder is a condition caused by a change or mutation in one or more genes or chromosomes. These changes may be small, a single letter in the code, or large, such as an extra or missing chromosome. They can be inherited from parents or arise spontaneously during development. Let us begin with single gene disorders, conditions caused by mutations in just one gene. These disorders follow predictable inheritance patterns. Some are autosomal dominant, meaning only one altered copy of the gene is enough to cause the condition. Others are autosomal recessive, requiring two altered copies, one from each parent. There are also X-linked disorders, which are tied to genes on the X chromosome and often affect males more noticeably, since they have only one X. A well-known example of an autosomal recessive disorder is cystic fibrosis. It is caused by a mutation in a gene that regulates salt and water movement in cells. People with two mutated copies may develop thick mucus in the lungs and digestive tract, requiring lifelong care and treatment. Carriers, those with one normal and one altered gene, are typically healthy, though they can pass the gene to their children. Another example is sickle cell disease, caused by a change in the gene that shapes hemoglobin, the protein in red blood cells that carries oxygen. The altered form causes red blood cells to become rigid and curved, leading to reduced oxygen delivery and episodes of pain. Yet this same mutation in carriers provides some protection against malaria, a striking example of how evolution balances risk and advantage. Some disorders are autosomal dominant, such as Huntington's disease. This condition leads to progressive changes in movement, mood, and memory, usually in adulthood. Because it only requires one altered gene to manifest, each child of an affected parent has a 50% chance of inheriting the condition. The gene is passed silently for decades, often before symptoms appear, a reminder of how heredity unfolds across time. Then there are X-linked disorders, such as Duchenne muscular dystrophy and hemophilia. These conditions often affect males more severely, as they have only one X chromosome. Females, with two X's, may be carriers experiencing no symptoms themselves, but capable of passing the gene to their children. In addition to single gene conditions, there are chromosomal disorders, which involve changes in the number or structure of chromosomes. One of the most recognized is Down syndrome, which occurs when a person has an extra copy of chromosome 21, a condition known as trisomy 21. This additional chromosome affects development, often leading to distinct facial features, mild to moderate intellectual disability, and a warm, sociable temperament. Other chromosomal disorders include Turner syndrome, where a female has only one X chromosome, and Klinefelter syndrome, where a male has an extra X. These changes may affect physical development and fertility, but many individuals live healthy lives with support and care. Some genetic disorders are not inherited, but spontaneous, caused by mutations that occur during the formation of egg or sperm cells, or in the very early embryo. These are called de novo mutations, and they can result in rare or unique conditions, even when no family history is present. Each case is a gentle reminder of the subtle unpredictability of biology. There are also conditions influenced by multiple genes, as well as environmental factors. These are called multifactorial disorders, and include many of the health conditions we encounter in everyday life: diabetes, heart disease, certain types of cancer, and mental health conditions such as depression or schizophrenia. In these cases, no single gene causes the condition. Instead, a combination of inherited tendencies, lifestyle, and life experience shapes the outcome. Genetic disorders are not always fixed in their effects. Some may be mild, with little to no impact on daily life. Others may be manageable, with medication, therapy, or lifestyle support. And some, though serious, may now be better understood and treated, thanks to advances in medical science. As knowledge grows, so does genetic testing, a tool that allows us to look into the code to see which alleles are present and to understand the risks or possibilities they carry. For some, testing provides peace of mind. For others, it offers a path to early intervention or family planning. Always, it is a personal decision, guided by compassion and understanding. Importantly, a genetic diagnosis does not define a person. It is not the sum of who they are, but one thread in a much larger tapestry. People with genetic disorders live rich, full, meaningful lives. They love, learn, create, and contribute. Their stories are not about limits, but about resilience, the quiet strength of adapting, growing, and thriving with what has been given. As you rest tonight, know that the human genome, though powerful, is not perfect. It is full of variation, of small shifts and gentle deviations. That is part of being alive, and within these differences, there is room for both challenge and beauty. You might imagine the genome not as a flawless text, but as a handwritten letter full of grace, but also little smudges and edits. Sometimes, those marks create something new. Sometimes, they ask for care. But always, they are part of the human story, and that story continues in every cell, every heartbeat, every breath. 17 Evolution and Genetics, the long story of change. In the stillness of night, when the present moment fades into silence, it becomes easier to sense the long flow of time, not just minutes and hours, but generations, centuries, and millennia. Beneath the calm of your breathing, your body carries a story millions of years in the making, a story written softly in the language of genes and shaped by the steady hand of evolution. Evolution is not a sudden transformation. It is not fast or loud. It is a quiet, ongoing process, one that unfolds through the slow accumulation of changes in the genetic code, passed from one generation to the next. Over time, these small changes can lead to new forms, new behaviors, and entirely new species. It is the natural unfolding of life's diversity, guided by variation, inheritance, and time. The foundation of evolution is genetic variation, which we've already explored. No two individuals are exactly alike, even within the same species. These differences arise from mutations, recombination, and random assortment of genes during reproduction. Some variations are visible, others lie hidden in the molecules of metabolism or the quiet workings of cells. From this variation, evolution takes shape through a process called natural selection. First described by Charles Darwin, natural selection is the principle that some traits make individuals more likely to survive and reproduce in a particular environment. If a trait offers even a slight advantage, better camouflage, more efficient digestion, stronger offspring, those individuals may be more likely to pass their genes on to the next generation. Over many generations, these advantageous traits become more common in the population. Meanwhile, traits that do not offer benefits or that hinder survival may fade away. The result is adaptation, a slow, natural shaping of species to fit their surroundings. But natural selection is only one part of the evolutionary process. Another is genetic drift, which involves random changes in allele frequencies, especially in small populations. Sometimes, by chance alone, certain alleles become more or less common, not because they offer an advantage, but simply because of random sampling. This drift can have lasting effects, leading to divergence between populations, even in the absence of natural selection. There's also gene flow, which occurs when individuals from different populations interbreed, sharing their genetic material. This mixing introduces new alleles into a population, increasing genetic diversity, and sometimes offering traits that enhance survival. Together, these forces natural selection, genetic drift, gene flow, and mutation shape the endless branching of the tree of life. From simple beginnings, single-celled organisms floating in ancient oceans, life has expanded and diversified, filling every corner of Earth. Bacteria and birds, flowers and fungi, whales and worms all share a distant ancestry, traced back through countless generations by the code in their DNA. Modern genetics allows us to see this history more clearly. By comparing the DNA sequences of different organisms, scientists can estimate how long ago they shared a common ancestor. The more similar the sequences, the more recently they diverged. This has revealed unexpected relationships, showing, for example, that humans are more closely related to chimpanzees than to any other living species, and that whales are descended from land-dwelling mammals. Even within humans, evolution continues. Though we often think of it as something from the past, evolution is an ongoing process. Over the last tens of thousands of years, human populations have adapted to local conditions, developing lactose tolerance in regions where dairy farming became common, or resistance to diseases like malaria in areas where they were prevalent. In colder climates, genes related to fat storage and body insulation became more common. In high altitudes, traits that support oxygen uptake were favored. These changes happened slowly, gently. Sometimes, evolution takes a more sudden turn through speciation, the formation of a new species. This can occur when populations become isolated from one another, either geographically or reproductively. Over time, as different mutations accumulate and selection pressures diverge, the two groups may become so genetically distinct that they can no longer interbreed. A new species has quietly emerged. This process has happened countless times, creating the vast diversity of life we see today. It is not driven by a goal or destination. Evolution does not aim to perfect. It simply responds to the present moment, selecting what works and discarding what does not, slowly, patiently, without intention. It's important to remember that evolution is not linear. It does not march forward, nor does it follow a plan. It branches, loops, and sometimes reverses. Some lineages flourish, others go extinct. The story of life is not one of constant improvement, but of endless adaptation. And though we often look at evolution on the grand scale, continents, epochs, extinction events, it all comes back to the tiniest shifts in DNA. A single mutation passed to a child, a slightly better fit for the climate, a quieter heartbeat in the night. Over millions of years, these small changes build mountains of diversity. Even now, your own DNA contains echoes of evolution. Genes passed from ancient ancestors who once hunted on plains, gathered roots and berries, crossed rivers, climbed trees. Some of your traits, the shape of your nose, your tolerance for cold, your ability to digest certain foods, are distant adaptations, softened by time but still present in your biology. And as you lie in stillness, that story continues. Your cells divide, genes are copied, tiny variations are carried forward, most invisible, many insignificant, a few with the potential to shape the generations to come. You are not outside of evolution. You are part of it, one chapter in a story that began long before you were born and will continue long after, written not in words but in molecules. So imagine, as sleep takes hold, the quiet unfolding of time, the long flow of genes through rivers of generations, the slow curve of wings and leaves and limbs, and within that, the soft pulse of your own inheritance, a blend of ancient memory and modern life. You are, quite literally, the outcome of evolution, shaped by it, held by it, and quietly carrying it forward. 18 Genetics in the future, what lies ahead. As you settle deeper into rest, your body wrapped in stillness, let your thoughts drift gently forward, not in haste, not with urgency, but like a quiet breeze through an open window. In the peaceful dark, we turn now to the future of genetics, where centuries of patient discovery are slowly unfolding into new possibilities. The story of DNA is ancient, but our understanding of it and our ability to work with it is still young. Only in recent decades have we begun to unlock the deeper layers of the genome, to read its messages, and to consider what gentle guidance it might offer in shaping the world ahead. At the heart of this movement is genomic medicine, the integration of genetic knowledge into healthcare. Where once doctors treated patients largely by symptoms and observation, they are now beginning to use genetic information to tailor care in more personal and precise ways. This approach is often called personalized medicine or precision medicine. Instead of one-size-fits-all treatments, doctors may one day prescribe medications based on your specific genetic makeup, knowing, for example, how your body is likely to respond, how fast you metabolize certain drugs, or whether a treatment carries unusual risks for you. In some cases, this already happens. There are cancer treatments now matched to the mutations present in a tumor, rather than its location in the body. Certain genetic tests can predict how you might react to anesthesia, or how effective a blood thinner might be for your specific biology. This kind of knowledge turns healthcare from something reactive to something proactive, helping people manage risks, detect disease earlier, or even prevent it entirely, all based on what is written in their DNA. Another area of promise is gene therapy, the idea of treating disease not by addressing symptoms, but by gently correcting the underlying genetic cause. In some cases, this might involve adding a healthy version of a gene where one is missing. In others, it may mean editing a faulty sequence so it works properly once more. While still in its early stages, gene therapy has shown promise in treating certain inherited conditions, like a rare form of blindness or some immune system disorders. Researchers are exploring whether similar techniques might one day help with more common diseases, like sickle cell anemia, hemophilia, or even neurodegenerative conditions. A powerful tool emerging in this space is CRISPR, a method of precise genetic editing that allows scientists to target specific parts of DNA and make changes with remarkable accuracy. CRISPR was inspired by a natural defense system found in bacteria. Now, it is being used in laboratories around the world to better understand genes, model diseases, and imagine new treatments. The idea of editing genes may sound dramatic, even unsettling, but in most cases, it is gentle and controlled, used to study cells, correct errors, or better understand how certain sequences function. Scientists are proceeding with deep caution and care, recognizing both the potential and the responsibility that comes with altering the code of life. Beyond medicine, genetics may also shape our future in agriculture, conservation, and even climate resilience. Through selective breeding, humans have shaped plants and animals for thousands of years. Now, with genetic tools, this process becomes more informed, helping create crops that resist drought, reduce pesticide use, or provide greater nutrition. In some cases, these modifications are already helping support food security in regions where farming is difficult. In conservation, genetic insights can help protect endangered species by guiding breeding programs, maintaining diversity, or even considering the reintroduction of lost traits that once helped animals adapt. There are even discussions, cautiously held, about whether extinct traits could be revived to help certain ecosystems heal. And in a changing climate, understanding how genes influence resilience to heat, disease, or shifting environments may help both humans and other species prepare for what lies ahead. Of course, the future of genetics is not only technical, it is also deeply human. As we gain more knowledge, we are invited to ask important questions about identity, fairness, access, and choice. Who decides what traits matter? How do we protect privacy in a world where genetic data can reveal so much? How do we ensure that discoveries benefit all, and not just a few? These questions are not easily answered. They ask us to move slowly, gently, and with humility, to pair knowledge with wisdom, to see the genome not as a tool to control, but as a story to understand, one that belongs to all of us. In the years ahead, it is likely that each of us will know more about our own genetic makeup. We may carry our DNA data on a card or in an app. We may one day help our children understand not just their heritage, but their biological potential. And we may do so not to control their lives, but to support them gently, quietly, wisely, as that future unfolds. The genome remains unchanged in its essence. It is still just a string of 4 letters, repeated billions of times. But in those repetitions lie the instructions for everything that lives, the quiet guide for cells, the silent inheritance of generations, and now, perhaps, a soft light toward what's possible. As you lie in stillness, think of the genome not as a destination, but as a companion, something that walks with you, shaped by the past, active in the present, and gently nudging the future forward. And remember that no matter how much we learn, no matter how deeply we understand, life remains a mystery in its fullness. Genes explain much, but not all. The way we love, create, forgive, and dream cannot be sequenced. The genome holds our biology, but not our soul. So rest tonight knowing that you are part of something timeless, a gentle thread that stretches backward through ancestors, forward through possibility, written in the code of life, carried in your every cell. You are part memory. "19. The Human Genome Project, mapping life's code, long before the first stars appeared in the night sky. The blueprint for life had already begun to form not in ink or stone, but in the soft spirals of DNA. For millions of years, it was copied, passed down, folded into cells, guiding growth and renewal in silence. And then, for the first time in history, humans set out to read it. This gentle quest became known as the Human Genome Project, a vast international effort to sequence the complete genetic instructions of a human being. It was a quiet, meticulous mission, one rooted not in haste but in curiosity, not in conquest but in understanding. The project began formally in 1990, guided by scientists from around the world and supported by public institutions and private researchers alike. Its goal was simple in essence, but profound in scale: to identify all the genes in human DNA and determine the precise order of the base pairs, the A's, T's, G's, and C's that make up the human genome. At the time, sequencing even a small portion of DNA was painstaking and slow. The human genome, with its over 3 billion base pairs, seemed almost unfathomably complex. And yet, through years of dedication, collaboration, and evolving technology, the task was completed ahead of schedule in 2003. The result was not a single genome from one individual, but a composite reference drawn from multiple anonymous donors, representing humanity more broadly. It was the first full reading of life's script, made available freely to researchers across the world. So what did the Human Genome Project reveal? To begin with, it showed that humans have approximately 20, 0 to 25, 0 protein coding genes fewer than once imagined, and far fewer than some other species. But it also revealed that genes make up only a small fraction of the genome. Much of the rest consists of non-coding DNA, once dismissed as junk, but now understood to play vital roles in regulation, structure, and timing. The project uncovered repetitive sequences, transposons, mobile pieces of DNA, and long stretches, whose functions are still not fully known. It offered new insights into gene families, evolutionary conservation, and the deep similarities between humans and other organisms. It also gave rise to entire new fields of study. Genomics emerged as the study of the entire genome, not just individual genes, but how they interact, how they are regulated, and how their expression changes in different tissues, environments, and stages of life. Bioinformatics became the bridge between biology and computing, developing the tools needed to store, analyze, and visualize the vast seas of genetic data. And comparative genomics allowed scientists to line up the genomes of different species side by side, revealing not only our closeness to primates, but our shared heritage with yeast, fish, and even the simplest bacteria. The Human Genome Project also brought human variation into clearer focus. With it came the HapMap project, an effort to map the common patterns of genetic variation, known as haplotypes, that help explain why individuals and populations differ in traits, disease risk, and drug response. These discoveries laid the groundwork for personalized medicine, ancestry testing, and deeper exploration into conditions both rare and common. And yet, perhaps the most lasting legacy of the project is not technological or medical, but philosophical. It reminded us that we are all remarkably alike, despite differences in appearance, culture, and experience. All humans share over 99.9% of the same DNA sequence. That fraction of variation, less than 1%, carries the details of individuality, but the shared code binds us together more deeply than we often realize. The Human Genome Project also taught us to respect complexity. It showed that knowing the sequence is only the beginning. Genes do not act alone. They are turned on or off in response to cues. They are influenced by environment, shaped by epigenetic marks, and embedded in networks more intricate than we imagined. Understanding the genome is like reading the notes of a symphony. The score is written, but the music depends on how it is played. Since 2003, sequencing technology has grown faster and more affordable. What once cost billions of dollars and took over a decade can now be done in days, for a fraction of the cost. Projects are underway to sequence millions of human genomes, allowing even more precise insights into health, ancestry, and evolution. But even as science advances, the genome remains a quiet companion present in every cell, invisible to the eye, and active in every breath. The Human Genome Project did not unlock every secret, but it gave us a map, a reference, a foundation from which to explore. You might imagine it now, the long ladder of DNA, stretching gently across your cells, thousands of genes, many still waiting to be fully understood, an alphabet of 4 letters, forming words, sentences, and paragraphs, some ancient, some newly written, a story not of one person, but of a species, and perhaps, too, you can picture the scientists patient and tireless working in softly lit labs, night after night, year after year, not racing, but walking steadily toward knowledge, and in doing so, giving us all a greater sense of who we are and how we are connected. Tonight, you carry that map within you, not in a book or a chart, but in the quiet of your cells. The Human Genome Project did not give you your genes, but it gave us the gift of understanding them, and through that understanding, a deeper appreciation of life's quiet design. 20. The story in our cells. A calm closing reflection. In the hush of the night, as your thoughts begin to soften and your breathing slows to a quiet rhythm, we arrive at the final chapter of our journey, not an end, but a soft resting place, a space to reflect on all that has been gently explored. Throughout this series, we've wandered through the architecture of life, through cells and chromosomes, through the double helix of DNA, and the steady steps of cell division. We've traced how traits are passed from parent to child, how variation shapes the uniqueness of every being, and how time and chance sculpt those changes into evolution. We've drifted through the soft language of gene expression, seen how epigenetic marks guide development, and touched upon the quiet possibility of future medicine. And at every step, we've returned to one central truth, that every living cell carries a story written in the silent folds of its DNA. Each cell in your body, from the skin on your fingertips to the neurons that dream behind your closed eyes, holds a full copy of your genome, a complete set of instructions, a memory of origin, a quiet potential. This code does not shout. It does not command. It whispers softly, steadily, with infinite patience. The story written in your cells is not random. It is inherited not only from your parents, but from countless generations before them. It carries fragments of their traits, echoes of their lives, and the resilience of ancestors who walked, breathed, dreamed, and endured. And yet, it is also uniquely yours. Your genome, while built from ancient parts, exists in a form never seen before, a new arrangement, a singular variation. The genetic instructions in your body have never occurred in exactly this way, and they never will again. You are not a copy. You are a moment of possibility, a living continuation of a story that began long before you and will continue long after. In this way, genetics connects us not only to our immediate families, but to all of life. The genes that shape your heartbeat resemble those of other mammals. The genes that guide your cell division echo those in plants and fungi. The basic language of life, DNA, is shared across all living things. This quiet unity, this deep biological kinship, binds together all of Earth's creatures in a shared inheritance. It's easy to forget, amid the busyness of daily life, that we are biological beings, shaped by unseen codes, built from molecules, governed by ancient rhythms. But as you lie in stillness, perhaps you feel it more clearly. The quiet complexity within, the slow pulse of biology at rest. Think, too, of the body's calm cooperation, of how genes and proteins, cells and systems work together with no awareness, no direction, yet remarkable harmony. Even in sleep, your body continues this quiet symphony, repairing, restoring, regulating, guided always by that soft genetic script. There is wonder in this, not the kind that demands awe or noise, but the kind that invites peace, that says, you are made of something enduring. You carry, within you, the memory of stars and forests, of ancestors and unknown futures. And in this knowledge, there is comfort, a sense of belonging, a reassurance that, no matter the day, the year, or the challenge, your cells remember who you are. They follow their script, they perform their functions, and they do so with quiet, tireless care. The story in your cells is not only biological, it is also symbolic. It reminds us that we are not separate from nature, but part of it, that our differences, though meaningful, are written in the softest variations, that even as we change, grow, and learn, we are held by a structure that is older than memory. So as this series comes to a close, take a moment to rest in that stillness. You have learned much about DNA and genes, inheritance and variation, evolution and epigenetics, but you have also remembered something deeper, that the body is a vessel of quiet wisdom, that the self is layered, and that science, even when complex, can bring calm. You may forget the terms, the sequences, the processes. That's all right. What matters is the feeling, the sense of harmony between the micro and the vast, between the cell and the cosmos. A reminder that life, at its heart, is not chaos, but pattern, not noise, but design. Let your thoughts now soften, like pages closing gently. Let your breath carry you further inward. There is no urgency, no need to retain, to study, to solve. You have already understood more than you know. The story in your cells continues, moment by moment, not needing words, just quiet presence, just continuity. And in the silence of sleep, the body listens to that story not with ears, but with enzymes, proteins, and structure. It is a lullaby older than language, more precise than any clock. You are not alone in carrying it. Every living thing does, from the smallest moss to the tallest whale, From the newborn child to the ancient tree, all life whispers the same code, with its own variations. A single message, infinitely expressed. Rest now. You are part of that message, softly written, deeply connected, endlessly continued.